Okay, so before I start with the class, so I upload. So first of all, you can start signing. I uploaded the slides to. You should be part of a shared folder. I created a subfolder and I uploaded the slides. I upload also some notes with the calculation we did in class, I think today. Just to know if you are aware of that, or if you, well, there is a folder, and in this folder there are the slides, and inside the slides are also references. So, yeah.
This is in the first lecture.
Huh?
This file was in the first lecture.
So there was a file uploaded to the website, but there is also a folder, a shared folder, which is called shared with participants. I'm not sure what is the way you get, you don't know about this folder. Okay. So I'll ask how you are supposed to access this, and I'll let you know, okay? But I should be able to do it today. Let's do this. While we're waiting for people, I'll write about. Okay. Okay. Now I think we can start. So, so any question about what we did last time? So we are starting again from the magic table, but any question from what we have seen? Something is unclear? Did any of you play with the arginine thing? The answer is no, because there was a mistake. And so if you had played, you would have realized. I can pretend I did it on purpose, but I didn't. And so if you do the calculation, actually, the equation you end up with for the protein synthesizing arginine is not what I wrote yesterday, but it's something like okay. So this is actually the equation. Okay. Good. So I think we can actually start. So what we are going to do now is to start again from this. So just a recap of the point where we stopped yesterday. So yesterday I told you one thing you can do is, and sort of imagining that you want to see what is inside a cell, the first things you do is that you divide the cells into the raw material that builds a cell. And for instance, you can start measuring what are the main components, and what you discover is that a cell is mostly protein and RNA, and most of RNA is in ribosomes. And then you measure this for one condition. You also understand that if you want to understand something, the easiest condition where to be is when you are in balanced exponential growth, because the internal composition is stationary. If you were not in balanced exponential growth, perhaps this quantity were depending on what was the initial condition, what was the history of the cell before the experiment, what was the exact point where you were collecting the cells. If you are in balanced exponential growth, you know that there is not this time dependency, okay? Or at least you can hope that. So what you do then is, okay, you repeat this table. and you want somehow to understand how this depends on the condition. And of course, you can plot, let's say, something about this as a function of a property of the condition, and the simplest property of the condition is the growth rate. And if you do that, you discover that there is a simple relation. So the ratio between the RNA content and protein scales linearly with the growth rate for fast enough growth. Now, there is this thing that sort of bends when you are at slow growth, but for now, we're going to neglect it. And for fast enough growth, what you see is that if you have very different conditions, you see that this ratio is about to be linear. It's linear with the growth rate. It's surprisingly linear with the growth rate. And what the point where I stopped last time was to say this is surprising because we know that picture that I will not show you again of the complex mechanisms of regulation and metabolism that determines actually what a cell does. Right? So for a cell, if there is glucose or if there is glycerol plus amino acids, it's a very different state. Right? It has to activate different transporters. It has to produce different molecules, different enzymes, and so on and so forth. So the fact that despite this whole mess, you get at the end a plot which is linear, it's very surprising. Okay? So what we want to do is to understand why. And so we can imagine that this is like, I mean, you can call this a growth law, right? It's, and this sometimes is referred to the first growth law. So it's something where you see clearly that there is a constraint in what a cell does, okay? And so whatever model you want to build to describe what a cell does, it has to reproduce this observation, okay? Very good. Okay, let me see what is, okay. Good, so how do we understand this? So you can start writing what you know about, sorry, this disappeared. You can start to write what you know about this system, okay? So the first thing you know is that if we look, sorry, no, I was missing a step. No, actually, this is what I want to do. So the first thing you know is that cells are growing exponentially, okay? So I talked about the volume. You can talk about the mass, the dry mass. So if I take my cells, right, and I take a milliliter of my culture, I remove water, I essiccate it, I weight it, what I get is something, and the weight of this is, Lorenzo, please, you distract me. No, I'm sorry. So you weight this culture, how much the bacteria that you have in 1 milliliter, and you get some mass. If you track it over time, if you are in exponential growth, you know that the rate of change of the mass is proportional to the mass with the growth rate. Is this clear? Okay. Good. Now, another thing that we know, and actually that is suggested by this plot, is that cells, to grow in mass, has to produce proteins. Okay? So a cell has to produce protein. How does a cell produce proteins? Well, a cell produces proteins by having a ribosome, okay, that it's reading mRNA and is sort of building a chain of amino acids that then will fold into proteins and taking like, well, amino acids which are attached to, well, it doesn't matter for this context, but it basically takes amino acids that are around and assembles into proteins, okay? So somehow, what we know, what we expect, and so I sort of told you before that so the majority of RNA is inside the ribosomes. So this is telling us something about the abundance of ribosomes, right? Okay. Do you remember that, or you want me to repeat it? Okay. So you want to be yes, you remember, or no, you don't remember. So one question is, what is this RNA and protein measuring? So most of RNA is inside the ribosomes. So ribosomes are made of amino acids, they're proteins, but they also have a structural component which is made of RNA. So this is like a big protein with many amino acids, but there is also inside the ribosome a structural RNA component. Okay? So when we extract all the RNA in the cell, what we have is some RNA is messenger RNA, some RNA is transfer RNA. Some RNA, most of it, what they extract, is the structural component of ribosomes. Okay? So when we look at this plot, when the ratio between RNA and protein is changing, what we expect is changing is the amount of ribosomes. Okay? So actually, how much ribosomes you have per unit of mass. Okay? The concentration of ribosomes. Is this clear? This is to say most of RNA, I mean, RNA is in many things, but most of it is in ribosomes. So what I expect is that if I go from here to this, if I double it, it means that roughly ribosomes are doubling, right? Okay? So inspired by this, and inspired by the fact that we know this very simple fact about biology, what we can imagine is that the rate at which mass and a cell grows, it's also the same rate at which ribosomes are able to make proteins. Okay? So that somehow the mass of a cell, or if you want the mass of proteins, I could even write, the mass of proteins, okay, it's equal to what? Well, the mass of proteins, what I expect here is that it's equal to some rate, okay, which is the rate at which ribosomes add one amino acid times the mass of an amino acid, okay, times the number of ribosomes that are active. So the number of ribosomes that are actively translating messenger RNA, okay? So what I'm saying here is, you imagine ribosomes add amino acids at some rate. The mass of proteins increases with a rate which is how many ribosomes I have around that are translating times the rate at which they translate, which is called the elongation rate, times the mass of one amino acid. Okay? This is called elongation rate. And for your reference, E. coli, this number here, the elongation rate, it's how many amino acids as ribosomes add per some unit of time, let's say per second. So just, I mean, I don't expect you to know, but how fast do you think this is? So how many amino acids do you think a ribosomes add per second? Let's say. You can say 0.1, 1, 10, 100, so scales of magnitude. Who thinks it adds, start from the top, 1,000 amino acids per second? 1,000. Who says 100? 10? Okay. 1? 1? Many people don't care. 0.1? Okay, many people don't care, are still weight, they are, okay. So the elongation rate for E. coli is about 20 amino acids per second, okay? It's not a number that is super easy to guess. One, well, okay, I'll skip, but I was just curious about how you could, what was your expectation. Okay, so it's about 20 amino acids per second, so it adds this number of things, okay? Now, this equation here, it's inspired, let's say, by what we have there, in some sense, but it's far from being something obvious, right? So I said, yes, I mean, it's true that ribosomes have to add to build proteins with amino acids, okay? And so it's true that if we increase the number of amino acids that is active, the number of ribosomes that are active, I increase the rate at which proteins are produced, okay? So the fact that it depends on how many ribosomes you have, it's okay, but it's not obvious that it doesn't depend on other things, right? So if I change, for instance, here, I don't say anything about the dependency on the pool of amino acids, right? How many amino acids I have around in my cell, right? So this number of amino acids could be larger or smaller, right? Here I'm sort of saying, I don't need to track it, okay? In principle, I should write it explicitly, right? And that number could change across conditions, okay? Another things it could depend on is how much messenger RNA I have, right? It could happen that in some condition I could have not enough messenger RNA, and so ribosomes are sitting there waiting for messenger RNA to arrive. Or it could be energy dependent, right? I could have conditions where I have enough energy so ribosomes can go at full speed, but I could have other conditions where this K changes and this condition dependent, okay? This is to say that here I'm writing this where the only variable is somehow the number of active amino acids, but there are other variables that could matter, right? How much energy does the cell have? How much amino acids does the cell have? How much messenger RNA is in the cell? How many transfer RNA are available to the cell, okay? So there are many, many strong assumptions. It's far from obvious that this is what you have, okay? The reason why I'm writing this is because I know somehow that if I change the growth rate, one big thing that changes is the number of ribosomes, okay? Questions? Okay. Now, the key point here is I'm thinking about the notation, 1 second. So this is the amount of proteins, but I don't want to call it P. Well, okay. Okay, well, let's keep. Okay, let's keep it P because then, I mean, it might become confusing. But, okay, so this is how much protein you have. Now, what you know is that we are in balanced exponential growth. So this means that whatever components I have, there is, I mean, if you are in balanced exponential growth, all the extensive quantity are proportional to each other, right? So we can express everything in terms of concentrations, and the concentrations are fixed. So what you know is that this MP, the mass of proteins, is proportional to M, right? Okay. So, do you agree? Okay. So if the mass of proteins is proportional to M, it's also true that the total mass dt is equal to K, well, let's say it's proportional to K, M, A, and R. Okay, yes?
Did you say that because the majority of the mass of the cell is the protein,
No, this is true no matter what. Okay. Now, this ratio here, it's big. I mean, it's like 0. No, it's smaller. It's 0.55, but it's true all the time, right? Because what we are saying is this ratio, right? I mean, what I'm saying is not that this is constant independent of condition. That if you are in a given condition and you have cells growing, if you are in exponential growth, they are always proportional to each other in exponential growth. Okay? So if you look over time, what I'm saying is if you look at log m over time and you are in exponential growth, you have this one. If you look at, so let's say this is the total mass. Here you have log mass of proteins. Okay? And R is the number, here it's the number of ribosomes that are active. Here? No, this is the mass of all the proteins inside a cell. And what I'm saying is, how do you add proteins? You add proteins by adding amino acids, right? By building a protein. And who builds amino acids? Ribosomes. We are in balanced exponential growth, and you see now why balanced exponential growth simplifies, right? Because then we can write the derivative of the mass as this one, okay? Okay? So now what we can write is that we can also write this thing as K M A over the mass, well, what is the number of active ribosomes is the mass of active ribosomes divided by the mass of a single ribosome. Okay, so the number is, I weight all the ribosomes and I divide it by the mass of a single ribosome. Okay, and so I can also write this as K MAA over MR, and then I have I can write this quantity here as phi R active times M. So what I have is that phi R active is the fraction of the mass of all the cells which are active ribosomes. Okay? Is it clear? So you have all the components of your cell, and you say what fraction of the mass of a cell is active ribosomes. This is phi R active. Okay? Is it clear? So now I can say that this is some, let's say, F times M. Okay? And so what I have is, I have that this one is simply F times this. So if I use this expression, I get F K M A M R times phi R active times M. Okay? But what I know is, so I just substituted this expression here. where I have the fraction of the mass which is proteins. Okay? I'm stupid. I didn't need to do this. Sorry, I didn't need to do this. Sorry. Whoop. So this is like, right. This is MP, okay. Right. Now, what I have is also that, well, all the components of a cell are growing exponentially with the same rate, right? So all the components of a cell are growing exponentially with the same rate. The total mass, the mass of all the proteins, or the DNA mass has to grow exponentially, right? Because the concentration is fixed. So if I equate these two equations, what I get is that if you want M R active over, so what I get is that these two quantities has to be the same, so this has to be equal to lambda MP. Okay? Right? Do you agree? Okay. So if you equate these two, what you get is that the mass of active ribosomes divided by the mass of proteins. Yes? Lambda is the growth rate. What I'm saying is all the components of a cell are growing exponentially, right? So this is true also for log MR, okay, which is a part of the proteins. So everything log DNA, everything is growing exponentially, right? Because we are in balanced exponential growth, so everything is extensive. All the extensive quantities grow with the same rate. Their concentration, it's fixed over time and depends on the condition. Okay, so if I put this equation together, I get that the active ribosomes, the mass of active ribosomes over the mass of proteins, okay, it's equal to some factor, which is, it's equal to lambda over K MAA over MR. Okay? Questions? So the math should be simple. Okay? What is the interpretation of what we found? So this is almost what we have plotted there. And what we found is that the mass of active ribosomes over proteins, okay, it's linear with the growth rate. Okay? Now, there are two differences with the plot that we are showing. The first one is that here we are calculating the active ribosomes. And the second difference is that here there is a non-zero intercept. Okay? So these two things, there is a simple way to reconcile them, which is inspired by the data, which is the fact that the mass of active ribosomes is equal to the mass of all the ribosomes minus the mass of inactive ribosomes. Okay? And so if we put everything together, what we get is that the mass of active of ribosomes over the mass of protein, let's call this R, which is what we are plotting there. I mean, it's not what we're plotting there. It's a proxy of what we're plotting there. So actually, let's say, this is MR over MP. It's equal to I have this term here, which I can put on the other side, which is equal to mR inactive over MP plus lambda over K MAA MR. Okay? So what the plot is telling us, okay? Okay. So I can rewrite this expression in a more interpretable way, but you see that this expression, what this is telling us is that what we have on the y-axis is something related to the mass of ribosomes, okay? And this term here, it's a constant intercept, and then we have this term where we understand what is the slope, okay? So let's take this and actually make it a little bit, no, not a little bit. Let's make it comparable quantitatively with what we have, okay? So in order to do that, what we have to do is to transform this quantity in the following way. So what we are measuring over there, it's the ratio between RNA and proteins. So it's RNA to protein. Let's call it R, okay? So what is this R? This R, how do I convert it into this? So what I have to do is that I have to say, okay, what is the amount of RNA that I have in a cell? Okay? The amount of RNA that I have in a cell is the mass of RNA that is, let's put it as a small number, the mass of RNA that is in a ribosome, okay? Let me think if I want to do it this way or the other way. Okay, no. So how do I get this number from the mass of ribosomes? Okay, so okay. Now let's start from this. So I have this expression here, and I have that the mass of ribosomes over the mass of proteins. I can write it as the mass of RNA that I have in a cell divided by sorry, something simple, but I'm getting confused. Right. So I want to write this. So let's write this. So I have the mass of ribosomes. I want to write them as the mass of RNA. This is the mass of RNA, and I put here the mass. This is the mass of RNA over the mass of RNA. Here I have mass R, and here I can put the mass of how much RNA is in ribosomes. Okay? So, okay. And then I have how much mass of RNA is in ribosomes to cancel this, times how much protein, how much proteins, how much amino acids, what is the mass of the protein part of ribosomes. Okay? And here I have the mass proteins, and here I have the total mass of proteins. Okay? Okay. Right, correct. Okay. Okay? So what I did is just to write the mass of RNA, of ribosomes, as the mass of RNA, and then I have these terms, right, which is, I brought them for the following reason. So this is like a simple dividing the mass of ribosomes into different terms, with the idea that these different terms, for instance, this term here, is not a property of the growth condition, or this one, it's not really a property of the growth condition. So this one, it's something which is structural about the ribosomes, right? So this is telling me how much proteins, I mean, if I take a ribosome, what is the mass of the proteins composing the ribosomes, and what is the mass of the RNA composing the ribosome, okay? So it's not something condition dependent. So it's something simple that, for instance, you can ask ChatGPT, right? So you can say, what is the mass of the protein part of a ribosome? It's thinking. Bacteria. Okay. Ribosome. Right, you can ask these two things, or you can browse them, and you will find an answer. Actually, you get it. So you get that you have this ratio, right? You get that the RNA, you have the total mass, and is done by RNA and by protein. So you know the ratio of these two, and you get whatever is 0.9 over 1.6. Okay? Now, don't do that at home. What you should really do is to go to, for instance, this very nice website, which is called Cell Biology by the Numbers, and it tells you everything that you want to know. Right? But what I'm saying is, these are numbers that are very well known. Right? They're very well known because people measure these things all the time. Right? So, and yes.
You assume that a ribosome consists of two parts, a protein part and RNA part,
Yes.
and then you divided its
Yeah.
mass to these two
Yeah,
parts.
exactly. Yes, exactly. Okay? Oop. Okay, so basically you can calculate these, and by calculating these and applying the same, you can convert what is the RNA to protein ratio into this factor here. Okay? So what you get is a conversion between the RNA to protein ratio with some conversion factor to the ratio between mass and protein. Okay? Actually, what you can do, even it's a little bit better, is to convert it with divide the mass of ribosomes by the total mass of a cell. Okay? And so I have this conversion factor. And so basically what you have is you have a conversion factor between these two numbers. Okay? How much RNA over DNA you have, and what is the fraction of mass which are ribosomes, which I'll call VR. Okay? Huh? This name, raw. Okay? No, there is no name. It's like yeah, it's some conversion factor. Okay? Which converts the ratio of the mass of RNA to DNA, to the RNA to proteins, to what fraction of mass is ribosomes. Right? The key point is that this conversion factor is condition independent, and it depends on properties of how the mass is partitioned into proteins and RNA into ribosomes, what fraction of yes, these kind of things, okay? So really something that is not a property of a growing cell, it's a property of proteins, okay? And good. Questions about this? So in some sense, the key point is this factor here, you can study it in vitro, so you can take ribosomes out of a cell, study how a ribosome is made, and calculate this number. Okay? What we are plotting over there, it's a growing cell, so it's a fundamentally different thing. Is this distinction clear? Okay, good. So this is a conversion factor, and so what we can convert is we can convert the growth rate, the RNA to protein ratio, which is what we're plotting there, to the fraction of ribosomes we have in a cell. Okay? And using this expression here, what we have discovered, I mean, what we have here, which is also what we see in the data, is that phi it's equal to some constant value, which is the fraction of the fraction of the inactive ribosomes, the fraction of mass in a cell that is inactive ribosomes, plus lambda over some parameter that I'll call kappa t. Okay? So what is this kappa t? Well, kappa t is something that is proportional. Well, there will be some conversion factor here depending on rho. Okay? But this factor here, it's proportional to the elongation speed, okay, times, again, some constant, okay, which is related to mass, to ratios of mass. So kappa t is proportional to the elongation speed, okay? So if you want, is equal to the elongation speed times some constant, okay? Where this is the elongation rate of ribosomes. Now, the important point is something I didn't tell you, but what is the black line? Okay? What is the black line? Well, you don't know this number here. Okay? We didn't know that this was constant. But we know that amino acids go at 20 the ribosomes go as 20 amino acids per second. Okay? So we know this K. And then this constant factor, it's, again, a ratio of what is the mass of an amino acid over the mass of a ribosome times properties that are structural properties of molecules, proteins, and factors inside a cell. Okay? So we know this number, okay? And the black line is the number that we know. Okay? So this number here, 1 over kt, kappa t, is the slope. Okay? Questions about this? Yes.
You wrote this equation in analogy with that equation that you wrote earlier, yes?
Yes, so I know that R is proportional to phi R. Okay? So I can always convert this axis here into another axis. Let's see if I have it here. Well, so I can always convert these, right, to phi R. I know that they're proportional. Okay? So if you want it, don't look at the right plot for now. Here, right, this is exactly that conversion. Okay? I know that conversion factor, so I know that there is some constant value difference between these two. Okay? The factor is roughly 0.75. Okay? So you take this number, you multiply, you divide it by 0.75, and you get this. Okay? No, you multiply by 0.75 and you get this. Okay? So it's a conversion factor. Now, the point is, you know what is this kt. Okay? So then you can say, how do you test this model? I mean, how do you test something, a model where there are parameters? You can try to find, I mean, you can say, okay, if this is true, right, I know that it's true for this value of parameters, I want to try to change the value of parameters, okay? So I want to try to find a system with a different value of parameters, okay? So one thing that you can do is you can say, I can take mutant cells, okay? So not the original bug that I was using, another strain, so another type of bacteria, I mean, the same species, the same everything, but with a mutation that makes ribosomes go slower, okay? So if I make ribosomes go slower, what do I expect? What is going to change here? There's only one thing that is going to change. K. So I'll make ribosomes go slower, and so what I expect is K to be smaller, but K is the inverse of the slope, so I should expect the slope to go up, right? And this is what you can do, right? So here you have the same plot for different strains, right? So these are different conditions. Now, the empty plots, the empty symbols with different shapes are different strains. You have 3 strains. Sorry, I'll go also here. You have 3 strains, okay, with different symbols. And these 3 strains have different ribosome elongation rate. So they have ribosomes that go faster or slower, that go slower than the original one, okay? And so what do you expect? Well, you expect this kappa t to be smaller, and so the slope to be higher, okay? And what you see is that the slope changes, okay? And now if you look at the slope, 1 over the slope, so 1 over the slope should be kappa t, and should be related, you can relate it to the translation rate, right? Because you know what is the translation rate of the ribosomes there, right? Now this is a completely different experiment, right? So here you take these bugs for 4 different conditions, you grow them, you look RNA to protein as a function of the growth rate. Then you do another experiment where you take this cell, you open them up, you take the ribosomes, you put the ribosomes out of the cell, and you measure how ribosomes elongate, right? So it's a property that is in a completely different world in some sense. It's a property of the ribosome. It's not a property of the full cell. Okay? And what you see is exactly what you expect, that the slope, 1 over the slope kt, is proportional to the elongation speed, okay? To the rate of translation in amino acids per second. And you can actually predict the slope. The slope is related to this conversion factor that you know. Okay? Questions? Yes. If you want, this is a result. Okay? It could, but it doesn't. Okay? It does a little bit, but you don't have a clear, I mean, if you look at this is the one with the slower slope, it's higher than this one, and then there is this one. So, let's say, as a first approximation, we neglect it. Okay? It could, it's true. But this seems to be constant. Actually, it's not fully understood why this is constant. Okay? Good, yeah? Why? What is the purpose of what? The conversion rate. It's because we measure something. We measure RNA to protein, and what we care about is, I mean, according to this simple model, is the fraction of mass which is ribosomes. That's the quantity that we can, I mean, when we write model, this is the natural quantity, but this is not directly measurable. What we measure is RNA to protein, and there is a conversion factor. Okay? Good. Yes? This one? Simply, okay, so this is, okay. So if you see that you have the mass of ribosomes, you can write it as the mass of RNA times this factor here, okay? And so you know all these three ratios, okay? And you know then what is the mass of ribosomes as a mass of RNA, okay? And then you divide it by M, okay, which is proportional to MP. And so if you divide it by, so then I can say mass of R over M, it's equal, which is PR, it's equal to the mass of RNA over MP, MP over M, and then I have all this term here, right? MR RNA over mass RNA, mass RPR over mass rRNA, mass R. Okay? And so if you look at this is exactly R, and this is exactly rho. Okay? And so this is 0. I mean, this is an adimensional quantity, and it's 0.75 around for E. coli. Okay? Yes. Sorry?
The mass of ribosomes in the M lowercase the first equation of
No, this one is the mass of a single ribosome. Okay. So you take one ribosome, you weight. Now, the number of ribosomes is the mass of total ribosomes divided. Okay. Okay, so what do you do next? Then what you can say is, okay, let's try to do, I mean, we understand these things of ribosomes, that they are related to growth, and sort of what we understood is the following. What we understood is that well, we understood that the mass over T is done by ribosomes. And, well, we understood that lambda, that phi r is equal to phi naught plus lambda over some parameter kappa t, where phi r is equal to mr over m. Okay? Now, ribosomes are one component of what is in a cell, right? But it's not the full component. It's a big component. If you look at these seems to go between 0.1 and 0.3, so it's a big part of the mass of a cell, but it's not full, all of it, right? So the idea is that we can sort of partition the mass into different components, right? We will have phi r and other terms here, okay? And these other terms will sum to 1, okay? So if you want, if you take the components of a cell in a pie chart, we know that there is one part which is r, and then there is something else, okay? And what we know is when we change the growth rate, we change the size of this slice of the pie chart, okay? So that this moves with the growth rate. Okay? So how do we understand? So we want to build the other parts, and sorry, let me add here that this is elongation speed. Okay? So we can try to perturb again translation with the idea of trying to see how the system responds. So what we did here, we did two types of environmental condition. One environmental condition is to change the growth rate by changing nutrient. Okay? So we have nutrient where you're slower and nutrient where you're faster. Okay? And then another, well, not really perturbation, but another consideration that we did was to compare different strains. So different types of bacteria with different mutant of the same bacteria with different types of ribosomes. Okay? We can do something else, and the something else we can do is to add some antibiotics to the culture. Okay? So what you can do is, why you want to do that? You want to do that because we understand that ribosomes are important, and you can add antibiotics that interfere with ribosomes. Okay? And so what we can say is, one way to change the state of a cell is to change the nutrient. Okay? Another way is to change how well ribosomes work. Okay? So the way this is done is that in the same way as you take your culture, you add bacteria to a culture and you grow them, what you can add to your food, to the food for your cell, is a little bit of poison. Right? You add a little bit of poison, and the amount you add determines how big is the effect. If you add a little bit, the effect is small. If you add more, the effect is bigger. Okay? And in particular, you add a particular antibiotics, which is called chloramphenicol, which interacts with the ribosome. So basically binds to ribosomes and make them not active. Okay? So you add that, and what do you do with this? You do exactly the same plot. Okay? So you take one nutrient here, and you start adding different concentrations of.
Excuse me.
Yes.
What is the purpose of adding antibiotics to a cell?
So we understand. So in some senses, we want to. We know that there is this relation with the growth rate. Okay. We understand this, and we understood this by changing nutrient. Okay. Now we want to change conditions. Okay. And the way we want to change condition is to, another way to change condition is to not change sort of the nutrient, but to change how well ribosome work. Right? And the idea is that I want to do it continuously. Right? So this effect here that we see proves that kappa T, it's really something related to translation speed, so that this model is promising. But it's not a changing condition. I use different strains. Right? Here I want to take one strain and being able to move the growth rate by messing up with translation. Okay? And the way to do it is to add antibiotics. Now, disclaimer, okay? Of course, sort of meta answer. Of course, answering this question, it's hard because it's also a question on, I'm building this with a logic, which is not the logic on which it was discovered, right? So there is also this component here. So just to break the wall between me and you now, okay? But the logic is also that you want to affect growth in a different way. Okay? So this is what you obtain. So this is the same plot where you have the previous conditions, okay? So no antibiotics. And now what you do is that if you add antibiotics, okay, what happens is, let's say you start from here, and then the numbers you get is the concentration of antibiotics you add at the beginning. So you can add a very, very little bit more, a little bit more, a little bit more. So if you add a little bit, what you see, focus on the x-axis. If you add a little bit, you decrease a little bit the growth rate. If you add more, you decrease a little bit more the growth rate. And then if you add here, you decrease even more the growth rate. Okay? So what we find is a completely different relation, completely different. A different relation from what we had before. Okay? So what we see is that now, when we change, there is still a pattern between the ribosomes and growth rate. But now it's still linear, but now with a completely different slope, right? In a decreasing way and not an increasing way. Okay? And so basically, if you want to describe these lines, you have two parameters, right? The intercept, when the growth rate is 0, okay, and the slope, right? So now, what you can say is that, this is, I don't know how convincing it is, but the intercept is, so this is the intercept here. It's not varying much compared to the slope. So most of the variation that you see is due to change of the slope, okay? So the way we're gonna write this relation, so this is like changing nutrients, let's say. This goes up with lambda. We have that phi r is equal to phi max minus lambda over kn. Okay? This is like an empirical relation that we see, and this is when it's adding antibiotics. Okay? And the idea here is that this phi max is constant. So this is constant, and this is constant. Okay? While this one, this Kn, is nutrient dependent. Okay? This is what we see empirically. Okay? Now, what this also suggests is that you never see, so somehow if you add chloramphenicol, so if you add these antibiotics that mess up with the ribosomes, you produce more ribosomes, probably as a response because whatever, you can make hypotheses. You produce more ribosomes, but then you never go over a certain value, okay? You never produce more than something, okay? So the idea is that if we want to build this pie chart, okay, what is the minimal hypothesis we could make? Well, the minimal hypothesis we could make is that there are two parts, R and some P, okay? Now, these two parts, okay, if this was the case, you can imagine of moving this one and this one, okay? What we see is that, well, in principle, phi R can go as, basically this can go up to phi naught, but if this was the case, you could imagine that this could increase to 1, okay? What we see here is not the case. So it seems that there is some incompressible part, right? That there is, let's say, P, and there is something, let's call it Q, okay? That at least we need 3 parts, okay? And we can imagine that we are changing the condition. So this is the environment, if you want, the condition. What is the condition in this context? The condition in this context is what is the nutrient we are considering, or what is the level of antibiotics we are adding, okay? So the condition is where we are in terms of, it's where we are in this table, right? What is the medium and what is the level of antibiotics, okay? So this is the condition. And what we have is that we need at least 3 parts. So we need some phi R E plus phi P, which depends on the condition, plus some incompressible part, let's call it phi Q, which is equal to 1. Okay? So we understand what is phi R. We don't know, we don't have an interpretation for these 2 parts. Okay? What we know is that, well, phi R of E plus phi P of E is equal to some phi max equal to 1 minus phi Q. Okay? This is what we know. And what we know is that whatever this phi P is, if we change nutrient, so if we change nutrient, phi P must be equal to phi max minus phi R, so must be equal to phi max minus phi naught minus lambda over KT. Okay. And if we change, so add antibiotics, what we expect is that phi P, right, must be equal to lambda over kappa n. Okay? So this is just by having defined the fact that there are 3 components, okay? And when we change condition, we know what one component does, and we say there is one component which is fixed and one component that varies. Okay? We know that the minimal model where, let's say, phi max is equal to 1 does not apply. Okay? Questions about this? Yes.
Sorry, so all that we are trying to model is related to the growing process. So it's not like that process involved to help us to understand the limitation.
What we are doing is to use, so we have this experiment, we understood that, let's say, what we see is ribosomes are a big part of the mass of a protein, okay, and behave in these ways when we add antibiotics versus when we add nutrients. Okay? So we are sort of building the model slowly. As I promised yesterday, we are building the model slowly from the data. Then I'll stop, and we'll start from the model and look at the data under the light of the model. Okay? Now what we are doing is adding one experiment at a time and using this experiment to constrain what we know about the data. Okay? So first thing we know is that the ribosome increases when we change nutrients, increase this way, and the slope is proportional to the elongation speed. So this has to do with how fast the ribosome work. Then we show that this plot, when we add antibiotics, ribosomes has a different relation, and the slope here depends on the nutrient. Okay? And then what I'm saying is, we have these three parts. Okay? We need at least three parts. And there is one part which are the ribosomes. And so when the ribosome increases, you need a part that decreases, right? Because the constant fraction, the pi, is fixed. Okay? So if you increase the percentage of one, something else has to decrease. And so the minimal model, consistent with what we know, is that there are three components, right? There is a component which is ribosomes, a component which is something, that I call P, which changes opposite to ribosomes, and a component which is fixed. Okay?
Please repeat the logic in which we arrive to the conclusion that there is a fixed Q.
Well, the fact that there is a fixed Q is because if you look at this expression here, right, in principle, you could imagine that these two, there is some condition where they're equal to 1. Okay? And you could imagine that this component here could go to 0. Right? That does not happen because you go to phi max. Okay? So in some sense, there is something which is always fixed. Okay? So, okay. Good. So now the question is how do you understand what is P? Okay? So we want to find proteins that behave this way, so that behave in this way here. Okay? And so the question is, what is P and what is Q? Okay. First of all, what do we expect to be Q? So Q, this sector Q, must be proteins, okay? So it's not just one protein, but it's a group of proteins for which the value does not change as a function of the growth rate. Okay? So it's something that is there and is always there at the same amount. Okay? So suppose that you want to have something that is there, so a protein that is there and is there always at the same amount, independent of the condition. Okay? Do you think you have to control it or not? You have to control it, right? It's something that needs to be regulated. So you need regulation mechanisms that is strong enough that always drives the amount of proteins to be at a fixed value. Okay? So this Q here, so the idea is that Q are regulated. So there is some negative feedback that always push this Q, whatever proteins are part of this slice of the pie, to be at the same value. Okay? Otherwise, you add one nutrient, you mess up with things, you will have different values. Okay? So the simplest things we can do to understand what is P, I mean, to see something that is P, is to look at, so one hypothesis is P are things that are unregulated. So you can start with, you can build inside a bacteria, you can have a promoter, so you can have a region that produce where RNA polymerase attach and starts producing the messenger RNA to translate that protein, that is always expressed, okay? So it's the opposite of tight regulation. It's something that you always express, and you always express at the same rate, okay? And so the question is, one simple thing you can do is that you can track the expression of one gene, let's say some protein. You look at the expression of one particular protein that is always expressed, okay? And so a simple way to do it is that this protein is fluorescent proteins, and so you look at how much light is shined by the bacteria. Right? The more this is expressed, the more you're producing, okay? And so you can look at one particular protein, let's say it's called beta-gal, which is actually related to lactose, and you look at the expression of this protein as a function of growth rate, okay? And if you do that, what you discover is that it has exactly the form you wanted, okay? So this is one particular protein that you express this way, that is constitutively expressed, that is unregulated. And what you see is that if you change the nutrient, right, you move over this line, okay? You move over this line, and you have exactly what you expect here, okay? If now you change the antibiotics, what you expect is that you move exactly on this line with slope 0, with intercept 0, okay? So these are proteins that are constitutively expressed, that are expressed in an unregulated way, okay? And you get this pattern, okay? Now, in terms of regulation, what you can do is that you can build another construct which has a feedback loop. So what you do is that you have some protein which is negatively regulated. So when it's produced, it stops being produced. And this is also regulate the production of these of the protein we are tracking. And if you do that, you always get the same value, okay? So this becomes fixed, okay? Yes?
So I guess when we're not regularizing all the protein in the cell, we're regularizing a particular protein.
Yes, this is just one protein. Just one protein. The idea is this sector here is not one protein. It's many proteins which behave, for which the sum behave in the same way. Okay? It's a sort of coarse graining of the content of a cell. Okay? So this is the moment where we collected enough information that we can go back and try to understand how to sort of understand this in a more theory, model, mechanism-oriented way. Okay? So this is what we have understood. So we have understood that there are 3 sectors, okay? Ribosomes, something else again. And these 2 something else differ because one is constant, it doesn't depend on the condition, is always the same, and this being always the same, it means that is tightly regulated. And then we have another sector which is other stuff, which behaves in the opposite way of ribosomes. Okay? And we can track the 2. So we have a way to track, to measure proteins that behave this way. So how do we build the model? And I'll start, and then tomorrow we continue. So let's say that, so what we know about growth? Well, we know that cells are growing exponentially. Okay. And then we know from the first, from this relation, okay, let's call it A, we know from A that lambda it's equal to kappa t phi r minus phi naught. Okay, this is lambda. Lambda times m. Okay? Now, we have an interpretation of what this equation means. Right? What this equation means is that this is equal to some kappa t times the mass of active ribosomes. Okay? And so this is proportional to some k, the number of active ribosomes. So what we are saying here is that mass increases in a cell because we have ribosomes that are translating proteins. Okay? And somehow the flux, okay, the rate at which ribosomes translate proteins is the growth rate. Okay? So here we have proteins. Okay? Which are built out of amino acids. And the rate at which proteins are built out of amino acids is, we can write it as kappa t phi r minus phi naught. Okay? Is it clear? Good. Okay. Then we can use this relation here. Okay. We know that there is this phi p. Okay. We can track it. It exists. We are not sure what it is. Now, we want to interpret it. So what is this phi p? Well, I can write here using this b. Sorry, this was A. This is b. What I can write is that lambda is equal to k n phi p. Okay? So, times m. Okay? The whole thing. So this is again lambda. Okay? Now, here, if I look at this equation, the first equation, what we know is that this kt is something that is a property of the elongation, right? We saw it. It's a property of how fast. This one, it's a property of how fast are ribosomes. Okay? Now, what is Kn? Where did Kn came in? Well, Kn came in when we look at this plot and we saw that these slopes differ. So Kn is a property of the nutrient, right? This is how we discover it over here. Kn nutrient dependent. Okay? So this is something that is related to the nutrient. Okay? So one simple way to put it inside this picture and to interpret this is that this corresponds to another arrow here, okay, which is how fast, well, the rate at which you convert nutrients into the building blocks of proteins. Okay? And the idea that if you are in exponential growth, these two fluxes are both equal to the growth rate. Okay? Yes? Yes? Yes, so this is, the confusion I think comes from the following fact, that we discover Kn by looking at the amount of ribosomes we had when we were adding antibiotics, okay? Then we did another step, which is to say there is this unknown sector P, okay? And what we said is, by definition of this sector P, it has to have this relation when we add it. And we also saw it here. So the fact that phi r decreased this way corresponds to the fact that this decreased this way, where the slope depends on the nutrient. Right? So the slope here is the same slope here, just with a minus sign. Okay? And so what I'm doing is I'm using this relation to interpret P. Okay? And the interpretation is that this is another flux. It's a flux that depends on the nutrient, okay? Because Kn is nutrient dependent. And so a natural interpretation is that this is related to the flux from nutrients to proteins, okay?
Excuse me.
You were perfectly synchronized, okay?
What do you mean by different nutrients are different nutrients that can build different proteins?
Here I'm saying empirically, what we see is that here we are changing nutrients, okay, and we have a slope, okay? So we see that when we change nutrient, we change this slope, so we change this parameter kappa n. When we add chloramphenicol, we move along this, but we don't change the slope, okay?
Yes, yes. I just wanted to know different nutrients build up different proteins.
No, I mean, different nutrients, I give glucose or I give lactose. I give different food to my bacteria. So for me, the conditions, so you see that all of this depends on the condition. For me, a condition is what food, I mean, in principle could be what food I give, at what temperature I keep the culture. Temperature is not here now, but could be how much, what is the temperature that I consider, how much antibiotics I put. These are all the conditions, right? It's the condition where I do the experiments. And change condition, you change growth rate.
So nutrients are related to the circumstances.
Yes, yes. Right.
Not to the cell.
Well, it's how this, I mean, it's how a cell responds to nutrients. But no, the nutrients, yes, are related to my choice as an experimentalist. Okay? Okay. So what we have is that basically we have these three conditions. So let me write them here. So what we have is the following. So what we have is that lambda is equal to Kt phi r minus phi naught, okay? And is also equal to Kn phi p. And then we have another condition, which is phi p plus phi r equal to 1. Okay? Now, to be more explicit, I could write these. All of these things are functions, or actually, sorry, this is equal to phi max. Most of these things are condition dependent. Okay? So if you want to write it explicitly as a function of the condition, I could say that phi r depends on the condition, right? So phi r can have a value wherever in this plot. Okay? So depends on the condition, plus phi p, which also depends on the condition. Okay? It's equal to phi max, which we are assuming we see that is condition independent. And here we have that lambda depends on the condition. and it's equal to KT, which in principle depend on the condition. If I change the growth rate, if I change the amount of antibiotics, I change kappa T. Sorry, I should not have said that. I mean, let's say kappa T depends in principle on the condition. Phi R depends on the condition. And also Kn, kappa N depends on the condition, on the nutrient. And phi P depends on the nutrient. Okay? Depends on the condition. So I have all this dependency of the condition, okay? But I have constraints. Okay? So I'll stop here, and tomorrow we will start from this. Okay? We will derive all the prediction we have already seen, in some sense, that are built in, and other prediction. But this will serve us as a framework to understand, sort of, to have a framework to understand physiology. Okay? Yes?
It would be nice that tomorrow we have a list of all the fees. P0, Pmax, P.
Yes, it's all here. Okay?
So
Yes.
is there a phi n? Because from nutrients to ribosomes, there should be a phi.
No, this is phi P. So our interpretation is that this part here, that is the part that is responsible to the flux from nutrients to amino acids, is phi P. Okay?
Because we have a phi r at the end, so it's from ribosomes to.
No, phi R is from amino acids to proteins, right? So this flux is saying what is the flux of that at which you produce proteins from amino acids? That is some number times how many ribosomes you have. What fraction of the mass it's ribosomes, okay? How fast, the interpretation is how fast you convert, what is the rate at which you convert nutrients to amino acids? The rate at which you convert nutrients to amino acids is how much proteins you have in this sector P that sort of are the ones responsible for metabolism. So from
Because the more nutrients, as you said, the slope changes.
yes, exactly. So the reason why we interpret this phi P and this rate is because we see this thing here, and so we have this natural interpretation, right? That somehow the growth rate is related to something of the nutrient, and as here KT something about how you make proteins out of amino acids, this is something that can be used to, yeah. Then of course, I mean, you can validate, let's say, you can validate these functional things in many other ways, which I'll discuss, but this is like a sort of starting way, right?
And the growth rate always starts from 0.
No, it depends. Eh? You can go very slow. It's not at 0, but you can, I mean, you see, you have points that are very slow here. But yeah, exactly 0 growth rate, yeah, don't grow. Okay? Other questions? Okay, good. So we'll see each other tomorrow. Bye, see you tomorrow.